First of all, I would like to thank the organizers for this opportunity to present some of our work here. In this talk, I will try to briefly show you why is it worthy to study natural products and mainly plant originated natural products, and through some examples of our own work, probably or hopefully I can show you why is it fun actually to do so. So plants biosynthesize a really enormous variety of different chemicals, and they do so in order to interact with, or let's say to adapt to their environment. So they actually synthesize molecules that attract friends. They want to repel foes, and certainly a lot of compounds are prepared for this. Of course, friends want to get closer. They want to have just more fun from this friendship. Foes don't really want to get repelled. So this is altogether a really complex and really complex communication that is going on between plants and their environment. And through the evolution, this communication network, let's call it like this, is constantly changing. So this actually leads to a situation where it is not an overstatement to say that whatever a plant biosynthesizes within this context, that has its purpose, and it will have some kind of biochemical target out there or in here, let's say. If we think about this, then it's absolutely no wonder that plant-originated compounds are really highly represented in approved drugs. This is a quite famous pie chart that shows the involvement of plant-originated compounds in drug discovery. And here there are a lot of compounds that are, well, either natural or natural product derived, or that took their inspiration from a natural moiety or a natural pharmacophore. And if we look at it, how much of them is actually unchanged considering its structure, then we see that plants don't really care our drug discovery initiatives. So they don't really care that we want to put their compounds into pills. They don't really care that we want to avoid certain side effects and so on and so on. So in fact, when we go to drug discovery, we typically need to apply some kind of chemical changes. And basically, this is what we are doing in our research group. We take nature as an inspiration, nature and natural products as an inspiration, and we try to navigate this chemical space and extend it in a way that we can hopefully find new bioactive compounds that have a high, strong perspective in terms of drug discovery. To this, we use actually natural product isolation and chemical synthesis. We combine the toolkit of natural product isolation that is typically techniques that deal with the separation of complex mixtures, typically representing the plant's chemical diversity, and we do chemical synthetic transformations of these compounds, which frequently leads to an increase of this already amazing diversity. And here are three compound families that I would like to briefly talk about a little bit more in detail. Well, first, these so-called steroids. These compounds have been discovered by the Nobel laureate Adolf Butenandt and his co-worker Peter Carlson. Well, Butenandt got Nobel Prize in chemistry not for the discovery of this compound and this compound family, but something quite related to these. And soon enough after their discovery, it came out that, that plants contain a really high diversity of these kind of compounds. 20-hydroxyecdizone is the most widespread of these, which is actually the active form of the molting hormone. And plants contain these compounds in a very high amount, orders of magnitude higher amounts than insects contain these. And their chemical diversity usually makes up for a lot of minor compounds present in plants. Here you can see a lot of possible chemical changes that can increase this diversity, so that right now we know 517 natural active steroids. Actually, we know more because there are a lot of new compounds still unpublished in our lab. These compounds are really fascinating for their complex ecological role. Certainly, I mentioned that they are the molting hormone, or analogs of the molting hormone in insects. That means that they are toxic in larger doses. And for plants, these compounds certainly mean some kind of chemical defense. So yeah, plant synthesizes active steroid, insect comes and eats it, and then well, gets sick and undergoes all kinds of problems. What is interesting is that these compounds have beneficial bioactivities in mammals, and actually also in birds, so in vertebrates in general. This bioactivity is first of all anabolic. That is interesting because these compounds are not hormonal at all, so we don't really know what is the mechanism. Some kind of adaptogenic effect, that means they are, let's say, something like ginseng, makes us faster and stronger and happier altogether. And therefore, they are also interesting because the same compound family with which the plant is defending itself from useless predators, let's say, it gives us benefits. So, for example, a cow can eat the plant and gets stronger and happier for it, while certainly spreading the seeds and so on. Now, going to this last aspect that I mentioned here, I would like to show you some results from one of our own studies that was a really interesting, had a really interesting outcome. A veterinary colleague asked me to start a collaboration because they found several songbirds to host ticks that are, well, kulancs in Hungarian, if you don't know. Ticks on these birds underwent some strange pathological way of molting. So they were molting on the birds. That is very rare because tick is a, well, shy animal. Whenever it changes clothes, it goes out of the host animal and tries to find some comfy hidden place where it can do that. So this is really unusual to be molting on the host, and therefore this guy's idea was that maybe these birds that are actually insectivorous, so they eat insects, probably they have active steroids in their blood, and then the ticks suckling on them get poisoned by that active steroid content. And indeed, when we tested some over 200 blood samples collected from songbirds, the songbirds survived, so no ethical issues there. The blood was collected properly, and just a few tiny drops were needed to this analysis. Well, a lot of plant-originated active steroids were found in their blood. So this is just a few examples. Over 20 different structures were identified in their blood. And the funny thing is that most of these compounds cannot be originated from the insect biosynthesis. So it must come from plants. But these birds don't eat plants. What eat plants? Well, caterpillars eat plants. Bird eats caterpillar. Tick starts suckling on the bird. Tick gets some serious problems. So this is a really interesting example how, for example, such a plant-originated compound can pass through the food chain and cause some really well, fascinating effect on the end. I mentioned this non-hormonal anabolic effect that these compounds can exert. Well, if one searches, just search Google for this compound name, and you will find a lot of companies that sell these compounds well, plant extracts containing these compounds in tons amounts, really tons amounts. I'm not sure if it's visible from the back, but this company, for example, offers 5 metric tons per month, and the minimum order quantity is 200 kilograms. Well, someone must pay for that, right? if they prepare it. And indeed, there are a lot of food supplements sold for this alleged anabolic activity, which in fact just led to the inclusion of these compounds on the World Anti-Doping Agency's watch list this year. So, in fact, they might be active for that. Well, that said, we were interested in the chemical composition of such extracts, and I have to tell you that being a natural product chemist is a very hard job. One starts grinding, let's say, a few kilograms of plants in the lab, and it is after a lot of chromatographic steps to purify interesting compounds, and after, on the end, you get with a milligram of something treasure. You are happy that it's a new compound, and then you ask a pharmacologist, okay, how much do you need for measuring this or that bioactivity for me? And then pharmacologist says, okay, give me 2 grams, 3. Of course, yes. So this is actually a very interesting and novel way that we started to do in our lab, that we purchase these extracts on really high, in really high amounts. So this is for 20 kilograms of this enriched extract represents a ton of plant material that otherwise would be really impossible to process in the lab. This 1 kilogram represents even more because it is pure. So, well, we ordered 1 kilogram of white powder from China. That I was curious how it will go out, but no one asked a question. They only asked who will pay the duty. Well, we paid it, so no problem. In any way, we are processing these extracts, and I have to tell you that the chemical diversity of these compounds that nobody has ever really looked into is truly amazing. Here I show you, well, some probably pretty scary hexagons if you are not into chemistry. But anyway, you can see that their structure is, well, everywhere where it's red is different from the original compound. And some of them, actually most of them are new and bioactive. Now here is just a little, well, summary of two of the most important bioactivities that we found for these compounds, or what we actually are currently exploring. Less polar compounds that we can get very easily from semisynthesis strongly decrease the drug resistance of cancer cells. They increase toxicity of chemotherapy, cytotoxicity, so the therapeutic side, not general toxicity. And other compounds that are these, well, let's say classical polar hydroxy steroid structure, these are more into the other side of the coin. They are anabolic, antidiabetic, and cytoprotective. And actually some of these compounds we just found recently within a very nice collaboration that they are really, really potent in protecting the blood-brain barrier, which will be the next talk about. Well, in the title of my presentation, I also had nanoparticles included. This is the only way we prepare nanoparticles, but this is a rather unusual nanoparticle preparation, namely that we conjugate our compounds with a squalene-originated side chain that is highly biocompatible, and this conjugate doesn't need any further additives. We just transfer it into water from its organic solvent, and then it nanoprecipitates, kind of forms nanoparticles on its own. And based on this idea, we prepared some hetero nanoparticles where we combined the compound that can sensitize cancer cells to a certain chemotherapeutic agent and that certain chemotherapeutic agent, namely doxorubicin in this case, and putting them together as some kind of a warhead to destroy cancer cells much more efficiently and actually in a targeted way. Here you can see some data of the activity. On this slide, I just show you another class of compounds that we are working on. These so-called paraquinol derivatives. These target a really fancy anti-tumor target. These compounds, namely the ATR checkpoint kinase, ATR and checkpoint kinase 1. This is a part of the DNA damage response machinery, and there are quite a nice number of active clinical studies right now aiming this therapeutic target. These compounds have their archetype in a natural product where these R groups are all hydrogens, and then now we are actually pretty, one could say, far away from the natural origin, but still the activity is the activity is being optimized and stronger. Now we are at the low nanomolar level of cancer cell killing activity, and we keep selectivity for the target and improve it actually. And the last topic I would like to briefly mention you is concerning antioxidants. Well, antioxidants are probably the most fancy compound group of natural products. Everybody knows about antioxidants, and they are so good and so healthy and save us from hell and everything. Still, there is a lot of problem with antioxidants, and whenever it gets to the clinical development, then it's really a mess. And we study much less well considered aspect of the bioactivity of antioxidants, namely that we consider these compounds as oxidizable prodrugs. Somehow most researchers in this field actually focus on the level of reactive oxygen and nitrogen species, that when their level is decreased in the cell, then it's good, because then well, the cell is protected from their harmful effects, and certainly that is right, and that is a very important, maybe the most important aspect. However, when that decrease takes place with the mediation of an antioxidant that scavenges said oxidizing species, then that antioxidant actually turns something else that stays there, and the bioactivity of these metabolites are very rarely considered, or is very rarely considered when we are talking about the complex bioactivity observed on the in vivo level. And we are focusing on the permanent chemical changes that result in really new chemical entities within this process, and try to take these structures as inspiration to build up something new. And here I can show you just briefly a few examples that we recently found. This is really the newest research project that we are working on. by applying biomimetic and actually biorelevant oxidative reactions, we get to a lot of very interesting new structures, and many of these exert very potent bioactivities. Well, I could go into details, but maybe if you ask some questions later, then I can answer that more in detail. Here I would like to just say thanks to all our collaborators, certainly members of my group and former members of my group, and also, of course, for the funding agencies and for your kind attention.